Welcome back to the Unit 3 lectures for MLT 110. This is part 2. In this lecture, we'll be covering blood banking, in particular ABO testing. Let's talk about the difference between qualitative and quantitative testing. We've talked about this in the past, but let's just remind ourselves. Quantitative testing is where you can report out a result as either positive or negative. So there's no number or value associated with it. And a lot of times we're talking about maybe a color change when we're doing testing. Again, it's just, is the substance there or is the substance not there, positive or negative? For quantitative, this tells you how much or the quantity of a substance is there. So there's usually a numerical value associated with quantitative testing. Some examples of quantitative testing would be our T cell counts. Remember, our T cells are our T lymphocytes, which are white blood cells. Also, drug assays could be quantitative, or our IgG subclass measurements. Remember, our IgGs are antibodies. Okay, so we have our qualitative testing and we have our quantitative testing, but we also have something that's kind of in the middle. It's called semi-quantitative, and this is more of an estimate of concentrations, and it can be used for antibodies. It can be done by making a serial dilution and then reporting out a titer. If you've ever heard of somebody's titer being low or their titers being high, this is a semi-quantitative test. So with quantitative, you're just looking for positive or negative, and quantitative, you're going to get an exact number and an exact value for the result. The semi-quantitative, you're going to kind of get a positive and a negative. Is the substance there or not? But you're also going to get a number value as far as a number range. So you're going to get, for semi-quantitative, you're going to get yes, it's positive, but it's going to be within this range. You're not going to get an exact number. So if you look over here at these 4 tubes in this example on the right hand side of the slide, you have 4 different tubes, and this was a dilution that was done, and 3 of the tubes, you can see down here, have produced a positive reaction. Let's just say this is an antibody test. The titer was high enough in these tubes, or there was enough antibody in these 3 tubes to produce a positive reaction. This 4th tube, there wasn't enough of the substance or antibody to produce a positive reaction. So that's a titer. One of the reactions that we look for in immunologic testing is agglutination. Agglutination is the visible clumping together of cells or particles as a result of a reaction with an antibody. Our IgM antibodies react best in agglutination reactions because of their large size. Remember, they're pentamers, meaning they have more binding sites or have a larger binding capacity. In most tests, a reaction of agglutination is going to indicate a positive test. If you'll look here on the image on the right hand side of your screen, this big old clump of red blood cells is showing agglutination. All these individual little red blood cells throughout this image are not clumped together, but you see several clumps of red blood cells on this particular image, and that's showing agglutination. On the image below, this is on a glass slide, and again, it's agglutination. This is actually an image that you're looking at agglutination under the microscope of red blood cells. This is agglutination you can see with your naked eye on the slide. And again, you're looking at red blood cells that have been clumped together. So how exactly does the agglutination reaction happen in a lot of test kits? Well, the agglutination reaction happens because Usually a lot of test kits come with latex beads in them, and latex beads are coated with a particular antibody. So here's what the latex beads look like with the antibodies on them. And then if the patient sample has the particular antigen that matches up with the antibody, then the antibody and the antigens will stick together or agglutinate. This image is to show you a comparison between a positive agglutination reaction versus a negative agglutination reaction. You can tell this one is positive because you have all these clumps in your sample, and you can see this with the naked eye. And over here the sample is very smooth. You don't see any clumps, so this is a negative reaction. So we grade the degree of agglutination on reactions. So this very first tube is showing you a negative reaction. There's no agglutination. Again, your sample looks very smooth. This next tube is showing you a 1+. You have very small clumps, but you still see, for the most part, the sample looks pretty smooth. You just have those small little clumps of agglutination. A 2+, you have very small clumps, but they're a little bit bigger than a 1+. The 3+, you have larger clumps, maybe 2 or 3 large clumps in a 3+ reaction. And then in a 4+ reaction, you just have one large pellet of red blood cells clumped together. And you can also have hemolysis in a sample. Remember, that's where your red blood cells have burst open and released their contents. That's why you have this pinkish color. In blood banking, we usually grade on a scale of 1+ to 4+, but it really doesn't matter if it's a 1+ agglutination or a 4+ of agglutination. Any reaction of agglutination is a positive reaction. There are 4 major blood groups in our ABO system, and they are A, B, AB, and O. There's a few different ways that we can perform ABO group testing. We can do a slide test, or we can do a tube test. These are both manual methods, or we can do gel testing, and this is an automated test. We're going to use the concept of hemagglutination for our blood type testing. This is where we're going to use our red blood cell particle or our red blood cells against antibodies to look for agglutination. And this helps determine the patient's blood type, and it's used for transfusion testing. Or in other words, help match a patient when they need a unit of blood. In blood type testing or ABO testing, we're going to use known antibodies, that's going to be our reagent, to detect unknown antigens on the patient's red blood cells. Okay, so our 4 blood groups, A, B, AB, and O, are all based on the presence or absence of antigens. And all these antigens are based on inherited genes from our parents. Antigens within our body can be found on several cell membranes like our red blood cells, platelets, white blood cells, and other tissues. But in blood bank testing, we're going to be looking for those antigens on our red blood cell surface. So what makes a person be a A-type person? What makes a person be a B-type person? And so on and so on. It's their antigens or their lack of antigens. So let's look at this image. So if you have a person that is in the A group, guess what? They only have A antigens on their red blood cells. If you have a person that is type B, their blood type is B, they only have B antigens on the surface of their red blood cells. If you have a person that is type AB for their blood type, they have both A and B antigens on their red blood cells. If you are type O, your blood type's O, you do not have any antigens. You lack A and you lack B. So you don't see any on the outside surface of your red blood cells. So again, when we're trying to test and determine a person's blood type, we're going to use commercially prepared antibodies or an antibody reagent to help determine or test what antigens are on the surface of the person's red blood cells. This process is called forward typing. Okay, let's look at the procedure for performing the forward type for the ABO reaction. The first thing you need to do is label you two test tubes. One needs to be anti-A, the other needs to be anti-B. Anti is representing antibody. So anytime you see anti, think antibody. I know it's a little confusing. You will see anti, you will think negative, but don't think negative, think antibody. You're going to need a bottle of anti-A, which is going to be the reagent with your antibody A in it, and you're going to need a bottle of reagent that's antibody B. This is going to have your antibody B. So next, you're going to add the patient's red blood cells, 1 drop, and then you're going to add 1 drop of the anti-A, again, antibody A. That's all you'll need in test tube 1. In test tube 2, you'll need 1 drop of your patient's red blood cells and then 1 drop of anti-B or your antibody B reagent. And then you're going to spin these down in the centrifuge and then you're going to look for an agglutination. So let's pretend we've performed the forward type reaction that we just went over in the previous slide on 4 samples. This will be our first sample, second sample, third sample, fourth sample. So let's look at our first sample. So what did we put in here? We put in 1 drop of patient red blood cells and we put 1 drop of antibody A reagent. In this tube we put 1 drop of red blood cells and 1 drop of anti-B or antibody B reagent. So now let's interpret our results. So in our anti-A tube, it looks like we have a negative reaction. We don't see any clumps. It's nice and smooth. In our anti-B tube, it also looks nice and smooth. We don't have any clumps. So both these tubes are negative. So what do we know? How are we going to interpret this? If we don't have any agglutination in either tube, what does that tell us? The antibodies in the reagents didn't have any antigens to react with on the red blood cells for this particular patient. So this patient's blood type is O because there was no antigens detected on the red blood cells. Let's interpret the results for the second patient we ran. Okay, for the anti-A tube, look what we've got. We've got a big pellet or a big clump of agglutination in our anti-A tube. Well, over here in our anti-B tube, we don't see any agglutination. It looks negative. So when we go to interpret that, what is our blood type going to be? It's going to be A because our anti-A or antibody A reagent was able to react with the A antigens that are on the red blood cells. That's why it clumped together. So this person's blood type is A. Now let's interpret our third patient sample. Let's look at anti-A first. Anti-A looks like a negative reaction. Anti-B looks like a positive reaction. It's a nice big clump, so this is a 4+ positive reaction. So if you look at all that together, it's going to be a type B person. This is because the anti-B or antibody B reagent is detecting B antigens on the patient's red blood cells. Now our fourth and final sample, it looks like there's a positive reaction in both tubes, our anti-A and our anti-B. So what does that tell us? That means it's detected antigens for both A and B on our red blood cells. Because remember, our reagents have antibody A and antibody B. So they have agglutinated with both antigen A and antigen B that are present on our red blood cells. So this person's blood type is AB. Antigens of ABO blood groups occur naturally in the body and are part of your inheritance from your parents. If you have a missing antigen on your red blood cells, you also naturally have an antibody for that missing antigen present in the body. Okay, so let's look at an example. What would that look like? So if you're a type A person, you would have A antigens naturally occurring on your red blood cells, but you would also have naturally occurring B antibodies in your body because you do not have B antigens on your red blood cells. Let's look at another example. Let's look at type O individuals. So type O individuals do not have any antigens on the red blood cells. So they would have both antibodies to A and antibodies to B naturally occurring in their body because they do not have any antigens on the red blood cells. In blood banking, we also can perform testing on our serum or plasma to test for the antibodies because that's actually where the antibodies are located, is either in your serum or your plasma, and that testing process is called reverse typing. So let's look at the testing procedure for reverse typing in blood banking. So our reagents for reverse typing are going to be actual commercially available red blood cells. You're going to have A1 red blood cells, and then you're going to have B red blood cells. Guess what's going to be on these red blood cells? A1's going to have A antigens on the red blood cells. Reagent B is going to have B antigens on the red blood cells. To actually perform the test, we're going to add to our first tube, our A1 tube, we're going to drop in a couple drops of our patient's plasma or serum. Then we're going to add our A1 reagent, which is our commercially available cells. Then in our B tube, we're going to add in some more of our patient plasma, a couple drops, and then our B cells. And then we're going to put those in a centrifuge, spin them down, and we're going to look for agglutination. And because this is the reverse typing, the results should be the exact opposite as the forward typing results. Okay, let's look at 4 results of reverse typing. And we're going to remind ourselves that the patient sample that we used was plasma or serum for reverse typing. The reagents that we used were A cells and B cells. Remember, these are commercially available cells, and the A1 cells have A antigens on them, and the B cells have B antigens on it. And remember, the results of the reverse typing should be the exact opposite of the forward typing. And we kind of use the reverse typing to confirm the results of our forward typing. Okay, so let's look at our first example here. You see a clump in our A1 tube, so we have agglutination. In our B tube, you have a clump of cells, so we have agglutination. So we have agglutination in both these tubes. So our reagent again was red blood cells with antigens on it. So this reagent that was used in this tube had A antigens on it. This reagent that was used in this tube had B antigens on it. Well, what did we need in order for there to be clumping? We needed antibody A and antibody B. So what does that tell us about this patient? This patient must be type O, because what's the only blood type that has both naturally occurring anti-A and anti-B in the plasma and serum. It's type O. And the reason they have antibody A and antibody B naturally occurring in their plasma or serum is because they don't have any antibodies on the red blood cells. Let's look at our second patient for our reverse typing. Okay, our A1 sample, it looks negative. There's no agglutination or clumping. But our B sample looks like there's a clump. There looks like there's agglutination. So how would we interpret that? It would be type A because it's the exact opposite of our forward typing. Remember, our type B has cells in it that has antigens, B antigens on it. So that tells us that this person must have antibody B's in their serum or plasma. So this person must be a type person. Okay. Third sample, you've got positive A1, negative B. So again, this would be the exact reverse of your forward typing result. So this would be a B type person. Our last example is negative for both our A1 tube and our B tube. So this person must be type AB, and that's because AB people do not have any natural occurring antibodies in their blood or serum. So we don't have any reaction with the A antigens in tube A1 or any reactions or agglutination with the antigen in tube B. So let's put everything together. Let's look at this chart of the forward and the reverse type together and ultimately what the interpretation of the blood type is. Let's ignore this column that says O cells. Let's ignore it for right now. But these first two columns are talking about results of testing for the forward type. These second two columns are talking about testing results for the reverse type. So we looked at all these samples already, but this is just putting it into a nice chart form for you. So a person that is type O, the forward results would be negative for the anti-A and the anti-B, but in the reverse type, it would be, the results would be the exact opposite. The A1 cells would be positive and the B cells would be positive. Okay, for an A person, again, the forward results are going to be the exact opposite of the reverse results. And it's going to be that same scenario all the way down on all four blood types. Your forward's going to be the exact opposite of your reverse results. And again, we kind of use the reverse typing in testing to kind of confirm our forward typing. The second most important blood group system next to the ABO blood group is the RH blood group, and it was discovered in the 1940s. And the RHD antigen is the most clinically significant of the RH antigens. RH antigens are highly immunogenic. This is why we must test a donor's blood in all patients who receive transfusions for the RH antigen. Also, RH testing has become part of the routine prenatal workup. Testing methods for the RH group can be done similar to the ABO testing. It can be done by a slide method or a tube method, which are manual, or the gel method. And here's an example of what the gel methods look like. And again, the gel method is an automated method, and it can be graded on the degree of agglutination, whether it's a 4+ or a 1+. But again, it doesn't matter if it's 1+ or a 4+. A positive reaction is a positive reaction. The Rh antigens are located on the surface of the red blood cells, just like the antigens of the ABO blood group. But if the person has the Rh antigens on the red blood cells, or if they're present, that person is known to be Rh positive or D positive. If they lack or do not have the antigens on the red blood cells, then they're known to be Rh negative or D negative. So to get a person's complete blood type, you must include the ABO group and the Rh. So an example would be A negative. The D antigen does occur in the majority of the population, with about 82% having the D antigen, but it does waver in as far as who has it between ethnic groups and racial groups. Antibodies to the D antigens are not naturally occurring in the body, but antibodies can be produced by the body if a person becomes sensitized or immunized to the D antigen. In a situation where a person that is D negative needs a blood transfusion, you always want to make sure to give that person Rh or D negative blood, because they can have a reaction to Rh positive or D positive blood. Another way that a person may end up building up Rh antibodies or D antibodies is in the situation of a mother, a D negative or Rh negative mother, giving birth to an Rh positive baby. This is why the Rh testing is part of the prenatal testing now, so that if you do have an Rh or D negative mother, we know it, and those D negative mothers can be given the RhoGAM shot so their body doesn't fight against or attack the baby if she ends up having, or the mother ends up having, a Rh positive baby. And if it's the mother's first birth and she's D negative and the baby's D positive, then the mother doesn't already have any antibodies to fight against the baby, so there might not be any issues with that pregnancy. But if the mother has a second pregnancy and she's obviously still D negative, but the second baby is again D positive, well, now the mother has built up D antibodies against the Rh. So then the second pregnancy, she may have problems because her body knows, hey, it's something foreign, it's D negative. So that's why we always want to test, and then that's why we give the RhoGAM shot, so the mother doesn't attack the baby, or the mother's body doesn't attack the baby and think it's something foreign. The testing procedure to test for the Rh D antigen is very similar to the forward typing in the ABO group. You're going to add a drop of the patient's red blood cells, and then your reagent's going to be the anti-D or antibody D reagent. And then you're also going to be running a control. It's going to be the red blood cells of the patient with a control reagent. And you're going to spin those down in a centrifuge, and you're going to look for agglutination. If there's agglutination in your anti-D tube, it's positive. It's a positive reaction for the Rh. And if there's no agglutination, then it's a negative reaction. So it's Rh negative. There is something called weak D. This is found in less than 1% of the population, and this is when the D reacts very weakly or even negative. These people, or these patients, are still considered Rh positive, even though they react very weakly or even sometimes negative. But there are other testing methods out there to determine if somebody is a weak D or not. There's a few other antigens in the Rh system that we want to mention. There's a C, or what's referred to as a big C. There's a little c, a big E, and a little e. Occasionally, these blood or antigens are tested for, but not always. If they are tested for, this reveals the patient's phenotype. You'll learn a lot more about the different blood groups and phenotypes when you get to your blood banking class, MLT 125. This image is just showing you a comparison of the Rh positive, the actual antigens on the red blood cell. It's also referred to as the Rh factor sometimes, versus the Rh negative red blood cell. You don't see any Rh or D antigens on this red blood cell. So this image is giving us an opportunity to interpret a blood type using the forward type and the Rh. So if you want to pause the video at this time and try to do it on your own before we get to the answer, you can. But let's go ahead and look at the result. Let's look at our anti-A. This would be our antibody A reagent. We can obviously see there's some clumping, so there's been agglutination here. Our antibody B, or anti-B reagent, there's also clumping. Well, our anti-D reagent, again, we're looking for the Rh factor. This looks nice and smooth. I don't see any clumping. Also, we're looking at our control. Remember, we run the anti-D in the control for the Rh testing. Well, your control for the anti-D or Rh should always be negative. So it should be always should be nice and smooth. If it's not, then there's a problem with your testing. So let's interpret the results. So if you got a positive here, what's our forward type? It's AB. So if we have a negative here and our control looks good, it's negative. So our answer is AB negative. Let's look at another example. This example is just missing the control, but we can still interpret the results. The anti-A and anti-B is for the forward, and the anti-D would tell us our Rh value. So the anti-A is clumped, so there's agglutination. The anti-B, there's no clumping, or there's no agglutination, so we would interpret the forward as A. And for the D, there's obviously agglutination, so this would be a positive reaction. So the overall result would be interpreted as an A positive. The next thing we want to look at is the recipient-donor compatibility chart. So let's say you are a person that has AB. So if your blood type is AB, that means you have antigens on your red blood cells of A and B, and you do not have any antibodies in your plasma or serum. So you can give blood only to an AB person, but you can receive blood from an AB person, an A person, a B person, or an O person. Okay, let's look at A. If you're a type A person, you have antigens on your red blood cells of A. You have antibodies of B in your plasma. And then you can give your blood to an A person or an AB person. And you can receive blood from an A person or an O person. Okay, if you are blood type B, you have B antigens on your red blood cells, and you have A antibodies in your plasma, and you can give blood to an A person or an AB person, but you can only receive blood from a B or an O person. And finally, the O blood group, you do not have any antigens on your red blood cells, but you have antibodies in your plasma to A and B. but you can give blood to AB, A, B, or O, and then you can only receive blood from other people with type O. This chart is showing pretty much the same information as the previous chart, but on this chart you can see a little bit better which blood group is the universal donor and which blood group is the universal recipient. The universal donor is an O negative person because they can give their blood to anybody. The universal recipient is a AB positive person because they can receive blood from anybody. I hope you've enjoyed all this information on blood banking, and I will see you over in part 3.